Eric,
Omar.
this is not a monster episode. This is indeed an anabolic episode, which might be, depending upon what we learn in this episode today, even bigger, maybe even stronger than a monster episode. Because joining us once again is the previous guest, all the way from 2019, Eric. It was crazy finding out that Alexander Kolliari-Turner, now PhD, Dr. Kolliari-Turner, is back on the podcast. For those that may or may not remember, there was something going on. There's kind of a natty Justice League forming all the way back in 2019. And the good now doctor, Alex, came on my YouTube channel, broke down some very interesting things on why it would not be feasible. And ever since then, it's been a very joyous meeting every single time we have a conversation. But Eric, I know what you did. It seems positive this episode. I know you did.
You always see right through me, my friend.
So, Alex, here's what happened. Okay, I'm just going to give you, I'd love to have you back on. Can't wait for you to discuss every single thing because truly, this is monster, next tier over, anabolic episode. Myself and Eric went and got tested, okay? Grant Tinsley, the Tinseltown's lab. We're going to wait to discuss the full findings when we have the good doctor back on. But I'll just say, Alex, there's something within our test results that raised some eyebrows. And I think what Eric's trying to do, Eric's stepping on stage. I stated in a private conversation with one Jeffrey Nipples that Eric has never looked better. That is a true statement, I said. And he said, wow. He said, really, in what way? But there were some things found, would you not agree, Eric, within our results, may or may not have to do with muscle mass and or bone density, that let's just say we're curious. And so we're looking not to exonerate ourselves, but just to have a full in-depth conversation about, anabolics, many different things of that nature, not related to our test results. Is that a fair statement, Eric?
I would say that this is a very heady conversation.
Yeah.
That, you're a headstrong person.
Yeah.
And so much to the degree that the DEXA scanner seemed to give results that would require someone to be like maybe 5 standard deviations over the norm for a certain metric. Now, I'm not saying I know for sure that anabolic steroids can make only your skull bone density extremely high. But let's just say I have looked at Omar differently these days. Yeah. And if we get in a fight, I'm not throwing head punches, I'm throwing body blows, and I'm absolutely not going to headbutt the man. That's all I'm going to say.
Alex, we are thrilled. Thank you for that, Eric, because we are thrilled to have you back on. It's always a great time. And you genuinely have some very interesting things to say in this episode. So we want to give you the floor. We have a ton of different questions, but please, for the listener now, it has been 4 years. Recently, is Lauren a comment? We're having like the victory lap, whereas 4 years later, for those that have not stayed for the 4 years or the new listeners we've recruited along the way, please, Alex, introduce yourself.
So my name is Alexander Kolliari-Turner. I have just finished a PhD at the University of Brighton in the molecular biology of anabolic steroids. The PhD research was funded by the World Anti-Doping Agency, and it was investigating this question of, does human muscle exhibit a memory of anabolic steroid exposure? And also, what is their influence on gene expression in blood, to try to look at a novel drug test for blood, and also what is their gene expression in muscle, to better understand how they interact with the androgen receptor and turn on genes associated with muscle growth. And in that part of the research, we used a novel technology, as novel as it had not been used in the anabolic steroid research before, called RNA sequencing, which sort of has been around since the mid-2000s now, which enables you to look at the expression level of every single gene in the genome, as previous studies have only ever been able to look at a handful. But there's been such an expansion now in next generation sequencing technologies that we had the ability to apply that technology now to human muscle tissue that had been exposed to steroids for the first time. So that was a project I've been working on. And both the gene expression results have now been published. They're published in May of this year, 2023. And our research regarding muscle memory and this concept of myonuclei permanency was published. It's published ahead of print. It's waiting to be signed for a journal article, but that was last year. That's been available as an abstract to be able to read online. And because of COVID, I also had to do some research to fill the time where I couldn't get into the laboratory and do these sort of, actual analytical methods. I've also published research related to the detection of steroids in the sport of weightlifting at the Olympics. And I've looked at what kind of drugs weightlifters have been caught taking over a 10-year period, what are the most popular steroids that have been detected. And also in terms of the Olympic Games, this idea of storing someone's urine at the time of Olympic Games, and then up to 10 years later, it's now 8 years, going back and looking at the frozen samples when the size has improved and seeing if you can catch doping weightlifters and doping medalists. And I've published a full analysis on that because in 2020, of all the years, turned out that the statutory limitations finished for London in 2012, 8 years before. And so you had the retesting of 2012, 2008, and 2004 Olympic Games all concluded. And actually turns out that more doping Olympians were caught via this sample reanalysis years later than were actually caught in real time at the time of the Games when the medals were actually handed out during that August period. And on average, it was about 6 years after the fact that these medalists were caught. And actually, it's the majority of medals that have ever been reallocated for doping at Olympics have actually been identified via retrospective, so reanalysis of samples. And so I've published research articles related to that as well. So that's my academic background. I know we're here today to talk mainly about the myonuclei permanency, the changing of the literature about does muscle exhibit a memory of resistance training in general, and then also this idea of can it exhibit a memory from anabolic steroid exposure, because there's this whole idea if you're caught taking steroids in sport, you have to have a 4-year ban if it's your first offence. If it's a second offence, it could be extended to 8 years or longer, depending on the severity of what your original ban was for. And so the idea is 4 years long enough? Is it not long enough? Shouldn't you be banning anabolic steroid users who get caught once or twice for life? And should we allow them to return to the drug-tested arena? Can you be a true lifelong natty if you've touched steroids? All these kinds of questions. So that's what we're here to discuss today. So I'm very excited to give you the findings from my PhD and now how the whole literature landscape has changed since I first appeared on your podcast all that time ago.
I'll tell you what, it's we're getting a lot of returning guests actually, Omar. Pretty soon we've got Cliff Wilson coming back on, and it's been a while for him as well. It's going to be a pleasant surprise that we've only burned a few bridges. I was sure that we would always be able to basically just have one and done with guests, but some of them don't dislike us too much to come back on. That's the praise that I will give ourselves. So Alex, I think it's awesome how far you've come. We met at the European Powerlifting Conference. I want to say the second year it was happening. Was it 2018 we met?
That's correct, yeah.
Yep, so that would have been in Dublin, and then the year afterwards, I did it again in London, and you finished your PhD. Now, you, like many of my students, have been in the same boat. You lost some years there, unfortunately, with COVID, but I actually think it's quite interesting that you were focused on the retrospective testing at the Olympics of weightlifters. And man, there's a lot that I want to cram into this episode because you're a wealth of knowledge and you're investigating a very interesting area. But why don't you take us where you think would be the best place to start? What do you want to cover first?
Well, I think myonuclei permanency is an interesting place to start because when I started my PhD, this concept of muscle memory was founded very heavily from research coming out of a laboratory spearheaded by a researcher called Christian Gunston, and he published a review paper in 2016 that basically said, muscle fibers are one of the few cell types inside the human body that contain many nuclei. The nucleus is the region of a cell that contains the DNA, and a liver cell, a kidney cell, a brain cell is a traditional animal cell. You've got one nucleus per cell, cellular body. But because muscle fibers by volume are the largest cells in the human body, they contain thousands of nuclei inside a single muscle fiber, being regarded as one cell in the body. And the idea with muscle growth is that you will, through resistance training, and or separately as well, and independent of resistance training through the intake of anabolic steroids, you will end up stimulating protein synthesis inside the muscle cell. And if that outweighs protein breakdown over a large proportion of time, the muscle fiber will increase in size. And eventually, the idea is that the nucleus inside the muscle cell, the myonuclei, can only support a certain area around it with proteins. Well, initially with RNA, which then will get converted into proteins. And eventually, there's going to be a ceiling where that single nucleus can't supply any more volume. And so the only way the muscle fibers ain't going to get any bigger is that you need to get more nuclei inside the cell. And the way that happens is you have these cells, which are called satellite cells, and they're called satellite cells because they're outside the muscle fiber, orbiting it, if you will, since their name. And you can activate them through persistent resistance training, and also through androgen receptor signaling with anabolic steroid usage, even in the absence of any resistance training. We know that from testosterone administration studies. So these satellite cells get activated, and then they progress into and donate their nucleus into the muscle fiber, and you then accumulate more myonuclei, greater propensity to make even more proteins, bigger muscle fibers. And the idea from basically seminal research papers where they did in vivo time-lapse imaging. So that means they're looking longitudinally through time, the exact same muscle fibers in alive mice. So you can think about that, how brutal that kind of studies on the animals. But anyway, they've imaged muscle fibers of mice, they've stained the nuclei, and what they've done is they've then forced the muscle fibers to shrink. So they've forced muscle atrophy. The muscle fibers have shrunk down in size, but the myonuclei don't go anywhere. They stay inside the muscle fiber. And so the muscle is then regarded as having a memory of being previously large and having high numbers of myonuclei. And that was the consensus of the field, is that myonuclei are permanent. And then in the context of steroids, that exact same research group gave female mice testosterone. Their muscle fibers increased in size. The number of nuclei went up. They removed the testosterone, and basically 2 months later, their muscle fibers shrank down to control levels, but the nuclei that were originally accumulated from the steroids still remained. And then when they exposed those mice to a resistance training stimulus, but actually in this context of this study, it was actually surgical hypertrophy. So they were cutting one of the muscles away from the mouse's leg, and then the mouse is forced to walk around the cage on a muscle, and that causes hypertrophy of that muscle. And basically, the mice that were previously exposed to anabolic steroids, because their myonuclei numbers were much, much higher at the point of that stimulus, their rate of muscle growth was much, much quicker, and their muscle fibers grew to a size that was 20% larger than the lifelong natty mice. And so there's this idea that myonuclei that get accumulated from steroids are permanent, and they facilitate enhanced rates of retraining and better muscle growth, even though more than 10% of the equivalent lifespan of the mice had passed. And so now the question is, in humans, are myonuclei permanent? And so at that point, that was regarded, yes, they're permanent. Look at all this consensus of this mice studies. And then it sort of basically turns out since then, there's been some other mice studies that have been conducted, and they've trained mice on a treadmill, and they've done progressive overload on a spinning wheel. So they've added weight to the wheel each week. The mice have been running on the wheel. They've been getting stronger and stronger. And they've had increases in muscle growth. They've had increases in myonuclei. They've taken away the training stimulus, and they've gone back and looked at those mice. And it turns out all some myonuclei have gone back down, and they've actually ended up losing myonuclei. So in the context of a more realistic hypertrophic stimulus, treadmill running, weighted treadwheel running in mice, myonuclei are lost. So straight away that puts a spanner in the works and saying, oh, okay, maybe they're not permanent. And then a human study was conducted by that original group at Christian Gunderson's lab. And in that study, they actually did also have a similar situation where they trained 19 subjects. Only 7 of them actually accumulated myonuclei from the training protocol, And 6 of them, their myonuclei in the detraining period all went back down to baseline levels. So even in human studies now, there's evidence in the context of normal resistance training that myonuclei can be lost. And so fast forward to 2020, and from the studies that took place, those 2 are notable ones, but there's other ones as well. A review paper gets written by Timothy Snyders and basically says in 2020, there is now no consensus on myonuclei permanency as a model of muscle memory. Now, muscle memory as a phenomena is real in a sense that we know that people who break their leg and their leg goes in a cast, they can regain their muscle mass. It's not like they permanently are going to have a smaller leg for the rest of their life. There is this idea that muscle mass can be regained, and you've previously that regaining time is much, much shorter than it took you in the first instance to build that muscle. And so that's the idea of the memory, is that the regaining phase happens in a quicker time frame than it was to accumulate in the first instance. So that is still an observed phenomenon. So what is the molecular mechanism for this happening if myonuclei may or may not be permanent? And so the field then starts to change to a different molecular mechanism of memory, of which a paper was published by the lead author, Robert Seaborn, and this was his PhD study. And it's in humans, and it's a detraining study. And although they don't look at myonuclei as a mechanism, it's done out of someone's laboratory called Adam Sharples, who's more interested in epigenetics. And basically the takeaway conclusion of that paper is that when you're exposed to resistant training stimulus, it will change the way your DNA is packaged inside the muscle cells, and it will change what genes are open for expression, and indeed also what genes are closed for expression. And so before and after the resistance training program, certain genes are getting turned on, and they're opening up for expression, and they're expressing their RNA, which eventually would be going on to be making certain proteins. And when you take away the training stimulus, those genes don't fully close for expression. They remain partially open, and then when you go back to training, they reopen again. And so your DNA, not the letters of your DNA, not the letters that of the A, T, C's and G's are changing. It's the methyl tags basically on the top. That's what we're looking at, is the methylation pattern of the DNA, the surface markers and that's in this context what they were looking at. And that would be regarded as an epigenetic change. So a change on the surface of DNA, and methylation takes it. And so that's got pretty good evidence that there is actually in humans an epigenetic memory of resistance training. So if myonuclei permanency in normal resistance training has got a question mark on it from my studies and human studies, but now we have this idea, there could be an epigenetic memory from human resistance exercise. The question is now, okay, so anabolic steroids, there's still this question mark in humans. Is there myonuclei permanency from anabolic steroid usage? So that's what we try to look at. And then now also, this question mark is, is there also a epigenetic memory of anabolic steroid usage? So I'll quickly touch on the epigenetic bit, and then we'll talk about the research I did. So the epigenetic memory is an interesting idea, because this whole concept here is that you're taking exogenous drugs that are binding to the androgen receptor across many cell types. That's why there's quite side effects associated with anabolic steroids, because the androgen receptor is present in your skin, your liver, your brain, etc. So it's binding to satellite cells, it's binding to the myonuclei, and a research study has been conducted where they've looked at the proteome. So, what proteins are getting expressed inside the muscle fibers of past anabolic steroid users. So the idea here is the drugs bind to the androgen receptor, that moves into the nucleus of the cell, it binds to the DNA, results in this transcription process that makes RNA, which eventually gets made into protein. And so that means that it's a theoretical possibility that they could change the way what genes are being opened and the way this DNA is getting packaged, because it's turning on these genes and making these proteins that are associated with enhanced rates of protein synthesis. And there was one study in past, very heavy exposure of anabolic steroid usage that has shown that the proteins that are being expressed in their muscle fibers is different to natural individuals, lifelong natural individuals, so at least in the context of that study. And so one possible explanation of that is that this androgen receptor binding has changed the way the DNA is being packaged inside the cells in a simplified manner. It's then changing what RNA is being made, and then it's resulting in a long-lasting change of what proteins are being made. So if they stopped taking steroids years ago, but the protein profiles are still different, there's a possibility that it could be from their steroid usage. And I say possibility because there's also a possibility it could be because they just have different training regimens and they have been training different muscle groups and different intensities and different styles. And that could be a big confounding variable as to why there are long-term proteome changes in those individuals who also happen to have taken anabolic steroids years ago for heavy dosages and prolonged periodic times compared to the naturals. So you've got one study, but it's in past anabolic steroid users and it's observational. So how much weighting can we put onto that? It's, just like my study that I did, it's observational. It's entry-level level research. I say that respectfully to what I did as well, but that's the reality. Is giving people anabolic steroids is ethically questionable in research. And so a lot of these studies are in this observational context. And so you have to interpret them with that level of investigation that has occurred.
Man, I tell you what, I'm more comfortable with you giving people anabolic steroids than some of the things that you said they were doing to those mice. So just call me an SPCA bleeding heart liberal, but that's how I feel.
You're not down for a little ablation? Well, what's the problem?
Synergist ablation. I tell you honestly, you can't see my soleus, but you can see my gastroc. So I've been thinking about this a little bit, but at least I get to consent. You know what I'm saying?
Yeah.
Well, the other interesting mice study, which I mentioned to you guys just before we started, which has also put a massive spanner in the works in all of this as well, is that they've genetically modified mice now to the point where essentially they insert a diphtheria, which I'm sure you know is quite a dangerous pathogen, a diphtheria toxin gene into the mice. And you can switch that gene on when you give the mice a drug. And you can genetically modify the mice so that gene only gets turned on inside satellite cells. And so that means when you give the drug to this mice, this toxic gene gets turned on inside satellite cells only and will kill all of the satellite cells in the mice at whatever time point in the life cycle of the mice you decide. So these mice are born normal. They have a normal upbringing in their cages. They grow to a normal adult size. And then once they're adults, you give them this drug. It will cause the satellite cell population in those mice to basically get deleted. Like 99.99% of their satellite cells just die because they start expressing this toxic gene only inside themselves. So that genetically modified mouse model has then been exposed to testosterone to see what happens if we try to then give the mice testosterone and what's their muscle growth potential like. And essentially, if you give those mice testosterone, even though they have no satellite cells and they cannot accumulate myonuclei, their muscle fibers will grow to the exact same size as regular mice that can accumulate myonuclei that have normal satellite cells. And so that means that the pre-existing myonuclei that were already inside those mice's muscle fibers, because they're adults when they're given this testosterone, have the ability to enlarge the muscle fibers to the exact same size as mice that can accumulate myonuclei and their myonuclei number go up. So that means you can obtain, at least in mice, comparable levels of muscle growth from testosterone exposure without any myonuclei accumulation occurring before. So that also begs the question, well, that means that in theory, we're hypothesizing here that humans could take anabolic steroids and maybe they don't get any myonuclei accumulation whatsoever and their muscle fibers will still grow, they'll still get large, but then there is no myonuclei memory effect if that is or is not true because they haven't actually accumulated any myonuclei from the steroid exposure. So,
Dr. Turner, that's incredible, and I got a question about that. Some of the speculative discussions I've seen in some of the papers on this topic suggest that accumulating more myonuclei is only necessary to continue muscle growth once there's been a certain threshold of hypertrophy achieved. That's kind of the whole point of myonuclear domain theory, right? Is it possible that in this study of mice that were set up to have diphtheria destroy their satellite cells, that they didn't hypertrophy enough to the point where they would have needed additional myonuclei? Or what is your thoughts there?
So my recollection of that study is there was no training stimulus. So it was chemical enhancement of muscle fiber size only. So maybe potentially, there's a certain chemical level of size you can achieve with no training, but then if you're going to train, the fibers want to get even bigger to adapt to that stimulus. And then, yes, maybe that could be a caveat that you're describing. But in general, Kevin Muir is a researcher who was involved in that research study. He's published some papers recently throughout the past few years where the concept of a very, very fixed myonuclear domain size, where there is like this number where as soon as you go above that number, that myonucleus can't support this area anymore. And once the fiber wants to get above that size, you have to get more to go in. It seems to be that there's much more flexibility in what is capable of a muscle fiber to hypertrophy itself without myonuclei going in. And so the transcriptional capacity of what a myonucleus is possible to do actually might be more than what we've thought of in the past, where it's not so much a very, very fixed threshold number. And his research is that there can be instances where there is like a reserve where the pre-existing myonuclei could actually just make up for it, basically, and could hypertrophy the fiber. And maybe we don't know what it is. Is it an energetic cost? Is it a trade-off at some point where the cell is having to make this decision of, do you want to accumulate more myonuclei and the cellular cascade that's associated with that, or could you just increase your transcriptional output of pre-existing myonuclei to compensate for this adaptation you're trying to achieve to facilitate what is happening to you? And at what point does it then have to cross the threshold where actually you're maxing out, you're like full throttle revving your pre-existing myonuclei, and maybe that's not good for the cell in the long run, and then you're going to have to accumulate more, and then so the time course spectrum and the flexibility of that area and that volume, I think it's a bit more flexible than was originally originally thought. But as to at what point can residual pre-existing myonuclei just sustain the adaptation and what's required, and at what point have you got to accumulate more, and at what point is the muscle fiber making that decision, and how is that decision molecularly being made? Very much an outstanding research question.
Yeah, that's a great answer. And one thing I was pondering as you were saying all this, Alex, is mice don't live very long.
Compared to us, obviously, and their fibers are also smaller than, by volume anyway, other mammals. So it can be maybe the overall volume of the cell as well. That is another caveat to how much can pre-existing myonuclei accommodate that total overall volume versus if your total overall volume, like if your room is much bigger because you're a bigger mammal and you've got just a bigger muscle fiber, maybe that number you are going to have to have a stricter number, and at some point there's going to have to be that integration.
Yeah, because I have no idea. Like my assumption is that the myonuclei in a mouse are probably a similar size to the myonuclei in a human. I literally have no idea. But if we're actually talking about different amounts of tissue, and then also the difference in lifespan in animals is something that I've always thought about, because generally the shorter the lifespan, the faster the metabolic processes. So it's kind of like they're aging at an accelerated rate. So when we think about the time scale in a mouse, how well that translates to a human is not necessarily clear to me. So those are those two very good points.
Caveats, as you said. Yeah, caveats, as you said. And so it is very much still outstanding research question of how fixed is a myonuclei domain. And review papers that are on it now saying there is essentially flexibility to it, and more research has got to be conducted to better understand how flexible is it really. As you said, how does that then translate into humans? Because this whole idea is that, the muscle mass getting lost through time in elderly individuals is a global health problem that sarcopenia, frailty, people falling down or being able to get out of chairs, breaking their hips, causing, emotional stress and burden on the individuals and also on the health services. And so if you could accumulate myonuclei in your younger years and hold on to them and potentially hold on to your muscle mass as you get older and be less frail, then that's the whole reason why muscle memory research is happening in the first place. Not you're a lifelong natty if you did one steroid cycle. Okay, that's interesting for us, but the original premise of this research was in that context. And so for humans, it's still an important research question that needs to be investigated for, those public health reasons.
Eric, two things. So first, Alex, to Eric's point, what you mentioned, the different lifespans, when you said it was for the mice, 10% of their lifespan, like think about the equivalent, right, for a human, that's what, 6, 7, 8 years. But secondly, Eric, just to contextualize this for the audience, myonuclei domain theory, some of the things that Alex is saying, why, if some of these lines of either research or some of these considerations that might challenge previously held assumptions, why it could potentially be seismic in the way that we understand things.
Yeah, it's a big deal. Our current kind of concept is you start training and you get increases in myofiber diameter, your traditional hypertrophy, and then as that grows, as Alex was saying, you need potentially more myonuclei to govern an area, because as the surface area grows, the volume of the muscle grows, the volume of the muscle fiber grows. It's more difficult for the cell to do its cell things when there is more distance between it and the next nuclei, because it's a multinucleated, largest cell in the body. All right, we got these satellite cells, they donate. And then once more myonuclei are developed, it's kind of this continuous process where then, okay, now we can hypertrophy a little more. And one of the quote unquote advantages of anabolic steroids is that they can take you past that, and that therefore also they will take you past not only the muscle fiber size you could have achieved without anabolic steroids, but also the myonuclei. And if you lose the size but keep the nuclei, oh shit, now someone who is a quote unquote lifetime natty has an advantage, right? And this has actually had, even just the preliminary concept has changed practice in sport to some degree. So for example, I know we've had Bob Bell on, head of the World Natural Bodybuilding Federation. He's talked about, their federation, how they operate. When I very first started bodybuilding, they had a 7-year drug-free policy where you would take a polygraph and they would, show you the banned substance list and discuss with you, have you taken anything on here? Okay, and it goes back 7 years. And they have some caveats in there. It's not all 7 years, like if you took a Fedra or something like that. Something that shouldn't have any lasting effect before you got involved in natural bodybuilding. There's some different time courses. They had amnesty periods for new over-the-counter pro hormones that come online because people might be taking them, like SARMs is kind of the new era of that, from a supplement store, right, or online. So now they have a 10-year polygraph. So I think that was directly influenced by some of these concepts. So that's kind of how it impacts our space. And it sounds like from talking to Alex that this is still an open question, and it's not that clear. And one thing that I've always been moderately critical of is that if we're just looking at the molecular stuff, but not actually looking at, but do they grow more muscle? Once we stop that cycle, are they growing faster? Do they get to a different endpoint? That's really important, because one of kind of the conceptual takeaways that I got from what Alex was saying, and this makes sense since we're only here because of many, many years of evolution, is that we have a ton of redundant systems, what we're built on top of. If we think about just the progression to be a human today in 2023, there's a lot of adaptation over time. And we've seen time and time again in physiology papers that, we think we found the pathway or the mechanism. And when we isolate it in an animal model, we remove it or we enhance it, it doesn't always do what we think it does. And sometimes people can still, get to the adaptation they're trying to. And we learn, oh, shoot, that's one of multiple overlapping systems that are there in place to do the same thing. And like Alex was saying, we can speculate. It could be, oh, wow, okay, the myonuclei are far more capable of upregulating their efficiency to govern this area. That's not fixed. So while it might prefer to activate a satellite cell, have it proliferate, become another myonuclei and govern more area, it can, if that's not an option, because somebody put diphtheria in there to, just simply make those myonuclei more effective.
And what you were saying there, Eric, was this idea, and in Gunderson's review papers, they speculate that myonuclei permanency makes evolutionary sense because way back when, if you had periods of lots of food and you were fed well and you could hypertrophy and chase down wild animals with your spear and feed and kill them, and then you then had periods of extreme famine where you're then going to lose all your muscle mass. Well, if your myonuclei were permanent during that time of muscle atrophy, when you were in the winter and starving, when you then go back into the following spring season, you can regain your muscle mass at a quicker rate because you've got pre-existing myonuclei that resided over that winter period. You could then get back into your fitness better, into your strength better, catch your food better, quicker, and that's an overall fitness advantage that would then be passed on down generations. And so it makes evolutionary sense that you have a memory of previously large muscle fibers in that context. But what is the mechanism of the memory? That's the outstanding question.
I mean, very well said by both of you. Alex, I want to jump back now because, Eric, you saw 2020, they basically tried to silence Dr. Turner because they were retesting certain samples, certain Olympic athlete samples, and it was causing a major stir. Then the pandemic. Talk to us about your PhD work, what you're doing, how you're trying to attempt to recruit participants in there, and then also you're saying there's something kind of a parallel going on. I think you mentioned off camera in the Netherlands. But I remember when you were trying to recruit that it was 5 part. Was that partially, Alex, due to once again just COVID and how it's tough, or also because the use of anabolics is illegal through a lot of the world? It's just hard to get participants. Talk to us a little bit about your research, what you were doing, and then catch us up to speed, Ben.
Yes. So we started the research study in 2017, and then we recruited right up until autumn of 2019. And we ended up having 56 research participants. And we ended up having individuals that we successfully managed to measure their muscle mass, 7 controls, 21 resistance-trained naturals, 19 current anabolic steroid users, and 7 previous anabolic steroid users. And then we endeavored to take muscle biopsies of those individuals, and of the 56 visits to the laboratory, 54 we get muscle mass measurements from, and 42 we successfully get a muscle biopsy for microscopy work, and 52 we get a muscle biopsy for the gene expression work. And the idea was, of those 19 current anabolic steroid users that we recruited, we were going to endeavor to try to monitor them through time after their anabolic steroid cycle to see how much muscle mass did they lose, and did their muscle fibers shrink, and what happened to the number of myonuclei. But then it ended up we only managed to do that for 5 individuals because then the pandemic happened, and it was illegal to get within 2 meters of somebody for months on end in the United Kingdom. And so it totally ceased sampling outright. We had sampling sessions organized, what would transpire to be a couple of weeks when we went, the United Kingdom entered the first lockdown in that March kind of 2020 period. And then since then, it became logistically impossible for us to basically recruit any more participants, which was a great shame. So at that point, I then couldn't enter the university buildings until late August. There's no way that I could have ended up sampling more participants. And so I managed to do some of the gene expression work in blood in the tail end of 2020. And then when we go into 2021, I then struggled to get equipment because this gene expression work that I'm doing, COVID is an RNA virus. I'm doing RNA sequencing, and the exact same reagents that I want to use are now scarce. And I had to wait months even for stuff like pipette tips, which is like the most basic laboratory thing if you want to do any sort of molecular work. So we then getting more delayed. And then because there's now a booming testing industry in the United Kingdom, it later transpires that the company we're using to do the sequencing gets sued by another company, and the machine that we're doing the sequencing on basically within a 12-month period, a patent infringement gets held up in the UK court, and that company, we can no longer use their equipment in the United Kingdom anymore. And so I didn't get to fully finish all of my experiments while that was happening. So then I had to send my samples to that company's headquarters, and they finished the research for us. And then I've managed to get the full muscle data set around Easter time of 2022. And then at that point, I then have the full data set. I've got all of the gene expression result for muscle, all of the gene expression results for blood. Thankfully, we'd already done the microscopy work in 2019. And then at that point, I then can knuckle down to do all the statistical analysis that's required for the gene expression work. And then my thesis comes around and I can hand it in the summer of 2023. So yeah, it was a long, arduous journey, but we eventually made it. And I suppose the results of the study that I conducted, and we're talking, I'm here the one talking to you today, but there's many people that helped in this research study. There was a fellow PhD student, Michael Giscard-Lima, who was integral in helping with particularly the myonuclei work, and he helped a little bit of the blood work, but I did all of the muscle gene expression work in the lab and in the statistical analysis in the end. But obviously my supervisory team, our collaborators that we had at other universities, and all of those people are referenced on the paper. So, but us as a collective, this research study was basically the first time that humans had been monitored through time after steroids, and that we'd had muscle biopsies taken to look at myonuclei numbers. And obviously all of us were disappointed that COVID happened, because it meant that our participant numbers were way, way lower than what we ever would have wanted to have had. As far as I also know, it is the only study really in like healthy men taking anabolic steroids where we've looked at how much muscle does somebody lose when they stop taking anabolic steroids. Like this comprehensive literature of steroid administration studies, notable, very famous testosterone administration studies that show how much muscle people can gain when they're given testosterone, super physiological levels of testosterone. But those studies never looked at how much muscle is then lost, and the rate of loss and the speed at which it happens. And so we have some base data on that in our 5 returning participants. And it's measured with a bioimpedance scale. That's not the best. It's not the DEXA gold standard that you guys would have done recently. But it's what we had access to. And that was what we ended up only being able to use. And so I guess I can give a little bit of update about what we found in that specific study, and then I'll tell you guys about my thoughts on the future field of how this kind of area of research could be improved, and then also some of the research that's being done in the Netherlands that's going to put even more light on how much muscle mass people lose after an anabolic steroid cycle. So so we'll talk about the loss of the muscle mass to begin with. So in our returning participants, 4 of them had finished taking anabolic steroids less than 2 weeks at the point when they first visited us. And then they returned between 19 and 28 weeks later. And this is an observational study. There's a range of the data they're returning, because I can't tell them, can you extend your steroid cycle a little bit longer so that you can come back, or any of those kinds of things. This is totally observational. It was beyond the ethics of what would have been allowed. They self-declared, this is also a caveat of the research, the level of steroids that they were taking. And we know that the vast majority of people are taking steroids that are made in underground labs, where there is no guarantee that what they think they are taking is what they are taking. Some of the research has come out of the Netherlands, I'll talk about shortly. Only about 40 to 50% of the steroids that they test in those studies actually contain the steroid that is written on the label. And so even if someone is telling me I'm taking 400 milligrams of X, I'm taking their word for it, but we didn't test the steroids and we don't know. So that's another future point for research in the future, is that we can't ethically give humans steroids. Well, it has been done in the past, but it's very difficult. It adds more expense, so the health monitoring that has to take place and the ethical implications of that. And so you're then left in doing observational research where you don't truly know how much drugs they're taking, what drugs they're taking, and if it's real. So sort of self-declarations. But they declared to us that they were using, on average, 505 milligrams of anabolic steroids plus or minus 236 milligrams for around about 8 weeks. So they're taking about roughly half a gram a year for 8 weeks on average, stopping around 2 weeks before they visit us. Then we muscle biopsied them and measured their muscle mass, and then they've come back somewhere between 19 and 28 weeks later, and they basically lost 3.9 to 4.7 kilograms of fat-free mass during that time period. How much muscle did they gain taking steroids? I don't know. So we didn't do the before the cycle. We only measured them at the point of cessation and then coming back 19 to 28 weeks later. But that's the first bit of data to see how much muscle mass, as measured with a bioimpedance scale, which is an asterisk of its accuracy, that was lost after a single steroid cycle. And on that point is where, during my PhD, I came across a group of researchers in the Netherlands who had conducted out of this city called Haarlem, and they called it the Haarlem Study, and it was all about harm reduction of anabolic steroid users. And they've managed to monitor 100 men before, directly after, and then I believe it said 3 months after, and then 1 year after taking anabolic steroids. And in that study, they did measure body weight, but they didn't measure muscle mass or do body composition testing. But they are repeating that study, and they're looking at body composition results to try and see how much muscle are the people gaining and then losing when they are coming off anabolic steroids. That study, in the terms of the health impacts, is very interesting because they have fine results that, for example, that even after one anabolic steroid cycle, there is a significant increase in some of these participants of the level of hypertrophy in their heart. So left ventricular hypertrophy is occurring even just after one cycle, but is then reversing when they come off. And they're getting aberrations in their cholesterol profiles, their liver enzymes, their kidney values, their blood pressure. And then even after just one steroid cycle, but then when they're coming off, they're all going back to baseline. And we're all getting, or the majority of those participants as well, I think it's something like 77% of the papers I was reading again today, their sperm count is dropping below what would be regarded as fertile after just a single steroid cycle. And in the majority of those participants actually didn't recover after 1 year full cessation of stopping, albeit, yes, maybe they could have done some things to try to accelerate that. And yes, those protocols could have been done, but that was what was observed in the research study. And so
pros and cons is what you're saying. There's
and literally one of their papers is towards the positive and the negative effects of anabolic steroids. All of the participants verbally declared enhanced strength in the gym, enhanced muscle mass, and so yes, there are both positives, but there are the negative health connotations. And just while we're talking about that, I guess the interesting thing is that I would say steroid usage now is probably the most prevalent it's ever been in society. And just in the United Kingdom, there's a professor based out of Manchester, a Metropolitan University called Jim McPhee, and he published a research paper with a group of researchers last year. And their academic, I say this respectfully, guesstimate based on survey data and their understanding is looking at a range of maybe roughly 500,000 anabolic steroid users in the UK of adults of aged late teenage years to early 60s, which they equate to being roughly something like 1 in 37 men taking anabolic steroids. And so we've got this data on the health impacts and what's happening even after just single steroid cycles, it is this idea that it could actually become a public health concern. And that's something that I know there are research groups in the UK looking at. And this group in the Netherlands, Diederik Smit is an endocrinologist who did this harm study. They've actually set up basically a harm reduction clinic that people can go to who are taking anabolic steroids, and they can have their cardiac function investigated, their hormone function investigated, their sperm function investigated, and they can know and find out the damage that's occurring to them. And then they can make educated and informed decisions about stopping, cessation, how to stop safely, and all of those kinds of things. But anyway, I'm sidetracking. So the data that will come off there, so that's the data from our muscle mass results. And then I think we will have much, much better data in the future about muscle mass losses, notably from that Harlem study. The Harlem study's not done any muscle biopsies. And so in our study, we did manage to take these muscle biopsies and count myonuclei in some of these individuals. And so I'll give you some of the raw data. For example, this is also where it gets interesting, and this is where you then get the caveats of having analysed the low numbers of participants. But just so people get an idea with a muscle biopsy, I mean, to do this, you have to inject a local anaesthetic into someone's, when we were doing the trapezius muscle, that's because previous research studies have shown the androgen receptors expressed at a higher level in the trapezius muscle than in leg muscles, even in naturals. And then in people taking steroids, that discrepancy is even greater. Anecdotally, this might be why, juiced up traps is a telltale sign. This is the bro science of someone taking anabolic steroids. But we were looking at the trapezius muscle. So you have to have a surgeon inject a local anesthetic into their upper back. They get cut with a scalpel, and then a needle that's about the same size as a standard ballpoint pen gets inserted. And then the window of the eye of the needle gets opened, and then it's manually closed. And you're then cutting out a piece of muscle that is about the same size as what would be at the end of a pencil eraser, or the rubber at the end of a pencil. And you're then covering that in a gel, like it almost got the same viscosity as like honey. And then you're dropping it inside liquid nitrogen, an alcohol that's been cooled by liquid nitrogen. So it's very, very, very cold to solidly freeze it. And then you're then mounting that into basically a very small, like, to say it crudely, like a salami slicer that's then thinly shaving the muscle fibers. So it's coming off and they're very, very thin. You're capturing that on a microscope slide. You're adding a variety of different stains, and then you're looking at the fibers and staining the myonuclei, and etc. So, for example, I'll give you an example of one of these participants. He self-declared that he was taking an anabolic steroid cycle for 8 weeks. This is one of the returning participants, participant 2. He self-declared he was taking 440 milligrams for 8 weeks. He stopped taking anabolic steroids 7 days before his first visit. He did conduct a post-cycle therapy, but self-declared that he was performance-enhancing drug-free the entire time. Turns out this guy's muscle fiber area actually increases, and it goes from 7,166 micrometers squared to 7,800 micrometers squared, even though he's returning 28 weeks after he stopped taking anabolic steroids. And his myonuclei go per fiber from 2.5 to 3.3. So he's actually made some gains, even though he stopped taking anabolic steroids. So that can be sampling effects, because I'm not sampling the exact same muscle fiber. The second visit, you're going back in and you're cutting out muscle based on where the needle resides. And so maybe that could be the reason for that effect. Or maybe he has genuinely actually managed to build some muscle because he's, still maybe got the ability to do that naturally, even though he has taken anabolic steroids. I'll give you another participant, returning participant 3, self-declaring he's taking 600 milligrams of steroids for 6 weeks. He's then actually, that guy has then, actually, I'll give you a better example. So participant 4, for example, is self-declaring 350 milligrams of anabolic steroids for 6 weeks. He's finishing his cycle at the point of his very first visit for us. And then he's then coming back. Oh no, sorry, that guy we didn't get a second sample for. So I'll go back to participant 3. So this last cycle, 600 milligrams of steroids for 6 weeks. He's then had 28 weeks between visits, and he has had a decrease in his cross-sectional area, and his myonuclei have remained roughly the same between visits. So although his muscle fibers have shrank, his myonuclei have remained roughly the same. So potentially that is evidence for myonuclei permanency, but you're looking at a very, very short time scale of usage. You're looking at a time scale of usage that is included within a standard ban in sport. So even if there is a memory, then that's not necessarily, you know, it would have been banned if that wasn't in the context of sport anyway. So the longitudinal data is not as good as we would have liked. I'll be frank with that, but it's the first concerted effort and attempt in the literature to try to do that. And then if we look at the cohort of past anabolic steroid users that we recruited, so they were guys that were self-declaring that they'd taken steroids to us more than 1 year ago, they'd stopped. Then their median usage of anabolic steroids in terms of duration of time was about 8 weeks. So they've self-declared to us that as previous anabolic steroid users on a median of 12 weeks of usage, and they've stopped taking anabolic steroids for, on average, 3.5 years ago. And their levels of myonuclei were exactly the same as a cohort of resistance trained natural individuals that we had recruited. So in that context, these guys had taken steroid cycles on average or median of a matter of a few months. They'd stopped 3 years ago, and then they returned, and we sampled them, and their myonuclei are not like sky, rocket high, and very, very large. So in that context, potentially some evidence there that in low scale usage in past anabolic steroid users who stopped years ago, their myonuclei numbers are not like more than what you'd expect to see in a natural training cohort. So that's what we found in our study. But there is another study that's very recently just come out, only like a few days ago, really, from us recording this, out of a group in Denmark. The two first authors are Nielsen and Rasmussen. And in that study, they've also recruited past anabolic steroid users and a population of lifelong naturals, same as us, that they managed to get 7 previous anabolic steroid users, and we managed to get 6. They had 10 controls, and we had 19 controls. And in their study, they had 4 years since cessation of stopping anabolic steroid use. And those past users did have a higher myonuclei density compared to the lifelong naturals that were recruited. And in that study, they saw that the longer that the self-declaration of usage was, the higher the myonuclei density of the past anabolic steroid users. And so potentially from this, we're looking at 2 observational studies here where we're recruiting past anabolic steroid users who are self-declaring to us how much anabolic steroids they took many, many years beforehand, of which in both instances of the study, we don't know if the steroids they took 4 or 5 years ago were real, and or if their memory is fully accurate as to what they were taking. Potentially there is this evidence that in my study, lower level usage is not resulting in really, really high myonuclei numbers. In this other study, potentially there is then this evidence that the more steroids that someone has self-declared that they've taken in the past, that there is a higher density of myonuclei compared to that cohort of resistance trained naturals that was recruited in that study. And then the final study of recruiting a cohort of past anabolic steroid users was done before we did our study. It's not published. It's someone's PhD thesis called Anders Eriksson. And that did find that the past anabolic steroid users in that study did have, only in their trapezius muscle, but not their leg muscle, significantly elevated numbers of myonuclei. But their level of usage was substantial. It was taking steroids for many, many years. Many years, and they'd stopped at least 1 year prior to sampling. But on average, I think in that study, they were self-declaring that they'd taken steroids for, on average, more than 5 years. I think it was in the range of about 8 years. So it's potentially, in past anabolic steroid users, say the current status of the literature of a small number of observational studies is that in our study, low-level usage doesn't seem in past anabolic steroid users to have had a high number of myonuclei. There's one study where heavy past anabolic steroid users do have higher myonuclei than recruited controls. And then there's this now recent study that's just come out of Denmark a few days ago that is potentially also suggesting that the more that someone is self-declaring that they've taken as a past user, the more that their myonuclei density is high. But then again, the biggest caveats are, we don't know, we'll recall self-declared anabolic steroid usage of years ago where people's memories naturally fallible. We don't know if the steroids they were taking was real. And then on top of that, there's the biggest confounding variable, which is the training regimens can be blurring all of these lines, and that you may be retaining a density of myonuclei because of the way you train harder, differently, more consistently compared to the other cohort of people that are recruited. And so the takeaway from my thesis is basically saying that I don't think, to fully answer this question, the field should be doing observational cross-sectional studies if you want to do this, because of all these confounding variables I've just mentioned. If you are going to do a cross-sectional observational human study, then you've at least got to somehow test the steroids that people are taking. But looking at a cohort of people who took steroids years ago and asking them, what did you take? How long did you take it for? Let's measure your myonuclei and compare it to people who are natural is ethically the easiest way to research this, but investigationally wise is not the best evidence way to go about it because of all these caveats and confounding variables. It's monitoring people through time, which is the best way for this to happen. And you can do that in an observational manner. I mean, look at the harm study. They managed to monitor 100 men before, during, and after a single anabolic steroid cycle, but they didn't take muscle biopsies. But if something like that was done, and you like they also did in the harm study, you tested their steroids, and then you found out were their steroids real, and you could actually see the actual dosage that the people were taking, then you remedy that confounding variable straight away, and you will get a much stronger signal to noise and a better experimental study design to be able to do that. And so we tried our best to monitor the people longitudinally, but we only had 5 returning participants. Of only 3 we managed to successfully biopsy through time, and the level of steroids they were self-declaring, the time scale of their returning frames and level, and when they were stopping the steroids was all different. So how much can we conclude from that is difficult, but that is the best way for it to be researched. But COVID curtailed our ability to be able to do that longitudinal aspect. But I really do think that the best way for the field to move forward to really answer that question is to do a longitudinal time course study, and to not do this recruit past steroid users, just look at their muscle and see is it different to natural trained people. That's been done now a few times. I don't know how you can't improve on that anymore. I think the best way to improve on it is for this commitment to the longitudinal aspect. So that's the findings, the myonuclei findings of us, the summary of that. Obviously, it's all going to be asterisks with the observational nature, but that's where I think the field should move into the future when it comes to this myonuclei aspect. And then, well, the other one is then also you naturally do get longitudinal time course study if you're looking at people who are transitioning gender. That is the other potential area where you could be looking at that and be a bit more standardized. But then we're talking about people who are going from natural circulating levels of testosterone of seen in cisgendered men to then that not being there after that's pharmacologically removed. We're then no longer talking about performance-enhancing testosterone, super physiological testosterone, synthetic anabolic steroids, which is more than we're interested in, about this idea of banning people from life for sport, from doping bans. So then it's a different research question. So
yeah, it's on one hand it makes sense, Alex. The greater the dosage, the longer the length of exposure, and then the longer the length of time away from that, the impact you'd expect to vary on permanence of myonuclei. And that makes sense. But I think quantifying it is very challenging in an observational nature. I think those broad brushstrokes, I wouldn't be surprised if those end up being true. But even just in the couple of participants you were discussing there, one of them might have been, juicing, but not past his own natural limit. One might have been. Another really common thing you see is that it can be quite demotivating to come off cycle, from talking to some of the guys who I know on the other side of the bodybuilding fence. And the training can sometimes get way worse.
Yeah.
But some of them, they're really, really objective about it. And they run, post-cycle therapy and creatine, and they do everything in their power to have like good post-cycle natty gains plus PCT. And they're able to, they do everything in their power to try to hold on to their gains and try to ramp back up their natural production of testosterone and other hormones. And, they do, and it's different. So it's a really challenging thing to say. And I do think training is actually a big spanner. I would assume someone who was training on gear and then stopped taking gear and stopped training might see more decay of their, myonuclei pool than someone who maintained training. So it is actually a lot of open questions in my mind, and it will be interesting to see the field move forward. And I think it's good that there's a little more openness in certain regions to kind of these harm reduction studies, because then you can get larger numbers. But I know it'll always be a challenge to actually administer anabolics in a RCT fashion, for sure, at least in most places of the world. The last thing I'll say is the whole like bunk gear thing is actually a very interesting topic.
Yep.
I had a friend of mine who is an IFBB pro, and I was talking to them, and I shared a story about someone I knew in my personal life who had thought they had run a single cycle, and it's a female, of Anavar. And I heard about this after the fact, and the only reason I heard about it was because when I met them again, they looked exactly the same. So I did one cycle, they came back, and it didn't stick around and make them permanently jacked. So there's another N of 1. You can add that to your data set. Just send me the Excel. I'll put that in. But their voice had permanently deepened.
Right.
And I thought I shared this story with my IFBB pro colleague, and they were like, to be honest, there's actually this huge issue right now where people think they're getting Anavar, but it's actually oral D-bol, which is far more androgenic. So that might have been what happened there.
And for a woman, it's particularly, and that
yes,
once your vocal cords and your larynx has changed, that is permanent. I've seen research studies of women who have taken nandrolone, so it's an injectable steroid, and after a few weeks, their voice has changed and altered permanently. And that's what the research study was, is a case study declaring that, so people are made aware of the risks in females that can occur.
Yeah, so it was just wild to me. It's like sometimes you think, oh, this is just, I'm gonna get these pharmaceutical grade things. I'm making an informed choice. So it's like, bro, you're buying it from a drug dealer who got it from somebody who might have made it in a bathtub. Like, let's be honest here. Like, this is not a clean transaction that you're doing, and they might try to upsell you to meth. That's the same person. So, I think some people underestimate the challenges of being in that world, and it always is a little bit wild to me as someone who's not part of it when I hear some of the stories.
Yeah. So looking at the Harlem study here, the specific anabolic steroid declared on the label was only detected in 47% of samples. They looked at 272 anabolic steroid samples from 46 different brands from the first 55 enrolled subjects of 100. And the specific steroid declared on the label was only detected in 47% of the samples. And 68% of samples undeclared anabolic steroids were found. So that means they were taking something that wasn't written on the label.
Sick.
And 49% of the samples contained a higher number of steroids than what was declared. And in summary, only 13% of samples were identified to solely contain the declared anabolic steroid.
Wow, that's
That
well. At least you get a little extra. I guess that's just the good customer bump, huh?
I'm giving you a bonus. Alex, I will say, bro, that's both a whole video. I've thought of this, that perhaps we'll do another episode. One, the existential, almost fundamental question, Eric, within lifting, like we're talking Olympic athletes or those at the top of their game, where now retrospectively how we can test it, how it changes our entire perspective. Like we know obviously it's a big issue, but that impact. The second one though, Alex, to your point, the amount of people, what they think they're getting and what they're actually getting, that massive discrepancy is probably one of the, if you want a scared straight episode of anabolic use, that might be one of the largest deterrents. I have quickly shout out to, we have a rascal athlete, his name's Carter Kissick. He's open. He was a natural individual. He decided to go on Hand School. Very open about everything, not in a way where he's trying to like sell it, but just saying, hey, and he had a disastrous like first cycle. We're speaking about potential outcomes. You're talking, Alex, like what is declared, what they think, and what it actually is. But it was the opposite, where there's something like harmful, where you had medical complications from it, and it was a light amount. Just talking about the high volatility, because these are, illegal compounds that you're purchasing that obviously aren't spoken about nearly enough. It is it's a little shocking, to be honest. I would say, though, if you want the opposite, playing the marketer, if you want the headline, Alex, for what you're saying about the research, is that anabolic steroids work. You just need to use them long enough to get more of the benefits. Those short-term people, they're using it and they found them, right? You need to keep using it. And then when you go off, well, then it's probably a better prospect. But no, it's I do view it, man, as real quick, because like I said, we could totally do another episode sometime. It's a fundamental question because people, I don't think they're only curious about using anabolics as a means of attaining a higher either physique or strength level, but it's within the domain of what is possible that they want to try and quantify. And there's always that ephemeral like other that people will attribute it to. I think everyone wants a hope on an outcome that is higher than what they feasibly might be able to obtain. Some will attribute that then to supplementation, the like kind of the fantastical notion that you could use supplements to achieve it. Or now, that's what I was joking with you, Eric, about, bringing back training hard, where it's this new mantra that gains, it's this a panacea actually of gains, that what you've been told, you just haven't been training hard enough and you can achieve it. So it's always this like fundamental question of potential, possibility, and then the individual wrapped up in all this that I find very interesting.
So, for example, in the Harlan study, where they monitored these 100 men taking this single anabolic steroid cycle, this is body weight, this isn't muscle mass. On average, their body weight was 88.8 kilos at the start of the cycle. They all take an anabolic steroid cycle as observational, so they were taking different dosages, different lengths, but they self-declared to do a single cycle and that they're coming fully off. And on average, their body weight at the end of that cycle was then 93.7. So you're looking at 5 kilo gain in body weight. 3 months later, this on average dropped down from 93.7 to 90. And then a year later, it's dropped down to 89.6. So they started the steroid cycle at 88.8. But before they started the steroid cycle, their body weight was 88.8. And then a year later, it was 89.6. And statistically, that was still significantly different, but there's only 1 kilo difference there in body weight. And so this is where you said the question is that you're going to take these substances that will result in an increase in muscle mass and strength, but health-wise, you can't take them forever. And you can, but there's serious risks associated with that. And you have to be very conscious and aware of them before you even think about it. And then if you then stop taking them, how do you psychologically deal with this loss in strength and muscle mass? And yes, you could do whatever you want, but there's still a reduction in actual like chemical stimulation of protein synthesis in your body that the drugs are doing for you. So even if myonuclei and you accumulate them from the steroid usage, in mice, you can still get muscle growth without myonuclei accumulation. Even if you do get myonuclei accumulation, and even if they are permanent, you're still getting this massive reduction in androgen receptor signaling and enhanced rates of protein synthesis, blunting of protein breakdown. That there's, chemically is what's going on with anabolic steroids. All of that is going away, and so the muscle mass reduction will happen. And to your point, Eric, if myonuclei are permanent, that you were saying before, what advantage does that then truly give the athlete? In the mice studies, we know it then causes them to regrow muscle at a faster rate, but we haven't had that in humans. We don't know if that happens in humans because the longitudinal study hasn't been done. You give the humans steroids, they beef up, you take the steroids away and they reduce their muscle mass, and then what you say to them, no more training, and then we're going to train you again and then see, do you actually build muscle at a faster rate? We don't know. We don't know. And in theory, yes, if you've got a higher myonuclei density, your propensity and your ability to recover, in theory, would be better. Your ability to actually grow muscle would be better. But does that actually really happen? There's no hard data in humans to even say that is true. Albeit, yes, it makes sense that you would believe that it would. And then hence why you have these longer bands in these natural bodybuilding federations, etc. But that actual real data in humans is not there. So,
well, Alex, I tell you what, it's a really interesting topic, and I can't thank you enough for coming back on, sharing it all, giving us a picture of not only what you did, but other labs, the future, the barriers, the challenges, and the open questions that remain. So, once again, just want to say thank you, and this has been a really good episode, Omar.
Alex, it is always fantastic to see you on Iron Culture. Hopefully not the last one. Don't think I didn't spot, by the way, my man, your forms are looking quite vascular. I could tell you've been putting in some of the work in the gym. I just thank God that we actually didn't talk about growth hormone and its potential relation to skull bone density. That's a whole other topic, right? Where that controversy we'll address at some point.
Remember, growth hormone is only detectable via a blood test. So if someone's only ever been doing urine tests.
And I have a fear of needles, so of course you can't test my blood.
Maybe by DEXA as well. DEXA might be able to pick up that. And maybe if someone's 5 standard deviations above the norm on their skull density.
It's naturally explainable, Eric. Five? Is 5 standard really, what is statistically, it's like incredibly rare.
Someone's got to be 5 standard deviations.
Someone's got to be, right?
Why not me?
Why not? So, someone's got to be 7 foot.
Somebody out there.
Someone's got to be 7'10". It's got to be me. But no, thank you so much, man, for coming back on. I want to thank every single person for listening to another episode of the Natural Podcast, Iron Culture. Yeah, we're going to declare that before every single episode. You can go ahead and leave a rating and review on iTunes. It does help us out. 5 stars is typically what people leave. We're back every single insert date here from now until the end of time. We'll catch you in that next episode.